Welcome to the lecture series on vitamins and nutrition. So today, as a continuation from previous session, we are going to look into various aspects of vitamin E mechanism of action, vitamin E biochemical reactions and the associated deficiency conditions. So today we are going to look into the aspects in which how vitamin E chemistry plays a major role in its mechanism of action. In the previous sessions we were looking at different aspects of vitamin E, and I told you like vitamin E basically is available chemically in different forms, but the one which we consider as nutritionally potent form would be the alpha tocopherol. I have been teaching you about different structural aspects of alpha tocopherol, and I told you that alpha tocopherol basically consists of this tocol nucleus, okay, and you have a side chain attached to it, and of all the forms of alpha tocopherol is supposed to be the potent form of vitamin E. So you may ask a question, so then what are these products? So tocotrienol and so on. So we have seen in the last session that all these products of vitamin E have their own function to play, but primarily the functional biologically active form being alpha tocopherol. So these actually occur as different versions or derivatives of tocopherol, but if you really look into the biological activity, alpha tocopherol is supposed to be the one with very good biological potency of more than 80% in mammalian systems. So these tocopherols do resemble the function of alpha tocopherol, but they may not be that effective because the way structural changes you find in them will decide the way they interact with different kinds of cellular components, including the receptors in which they act. Now in the previous session we looked into various aspects of vitamin E nutritive sources, and then we looked at different aspects in which how vitamin D is transported across, and now today I am going to explain little bit more on the mechanism of vitamin E. So if you look at vitamin E, in the last session we were looking at the antioxidant effects of vitamin E. So today I am going to elaborate more on the antioxidant effect of vitamin E, how vitamin E is said to be a potent antioxidant. In fact, the very role of vitamin E in variety of biological functions being its antioxidant potency. So in that way you could consider vitamin E function to have antioxidant effect in a native biological setup and also in the formulations in which vitamin E is used. So this aspect I have discussed earlier. So today our focus would be more on looking at the mechanism of tocopherol. So you look over here, tocopherols function by donating the proton atoms to radicals. So if you know what are free radicals, so whenever there is a cleavage of a bond, especially for a covalent bond, if you consider, the cleavage can be homocyclic or heterocyclic. So a heterocyclic cleavage of any biological chemical species would give rise to a nucleophile and an electrophile. But if it is going to be a homolytic cleavage where the donated electrons are being withdrawn by those corresponding components of atom that partake in that particular formation of a compound, they retain back that one electron. So these species, what we call them as free radicals, result from homolytic cleavage. Now you see here, this is an example of a free radical. So this black dot represents the electron that it has kept within itself, which was earlier used as part of covalent bond formation. So now what is the function over here? Now this is let's say a free radical generated as a byproduct of cellular metabolism. So this free radical is available as a byproduct, and now you see over there this tocopherol having a proton, and this free radical basically attracts this proton. So this proton is taken up by this free radical. So you see over there the corresponding proton bonded with the free radical, and now this is a neutralized atom. So now you consider this neutralized compound, which was originally a highly reactive free radical, has now been neutralized. Neutralized in this sense, I would say, I mean by their participation in a chemical reaction has come down. So they are no more reactive. Whereas what happened to this ring system? So this ring system has now generated a corresponding free radical. So this phenolic free radical, as we call it, this ring, has an advantage because it can be stabilized. So you see over there benzene ring with the resonance ring pattern present over here would provide the kind of stabilization, the pi electron cloud would provide the stabilization required for keeping this free radical in check. So you mean that this free radical would be far less reactive than this free radical. So what we call this as tocopheryl radical. So this radical, because of the presence of benzene ring, is relatively unreactive or least reactive. So what do? They get converted. So it reverts to tocopherol by a redox reaction with a hydrogen donor such as vitamin C. So that's why I said wherever you are going to study about antioxidant compounds, you always study them in a very close association with each other. So you would always get help, partnership from the other antioxidant to keep the cycle going. So in this case, tocopheryl radical would be converted back into tocopherol by a redox reaction with the hydrogen donor such as vitamin C. So if you look at it closely, you would be able to understand that this free radical, which is basically tocopheryl free radical, which was extremely reactive, is being converted into a relatively unreactive free radical, and this answers the question of antioxidant effect of tocopherols. And now this free radical in turn would be regenerated by one more antioxidant, and that's the job of vitamin C. So when we are going to study about vitamin C under the category of water soluble vitamins, we will see elaborately what would be the function of vitamin C in different tissues. Right now I want you to be aware of how the vitamin E gets replenished of its antioxidant activity by the presence of these helping antioxidant compounds like vitamin C. So you see over there tocopherols are incorporated into the cell membrane. So which site of the cell is more prone for oxidative damage? Obviously the outer surface, because this is the surface where you always see them in a dynamic mode. What do you mean by in a dynamic mode? So it means there is constant activity, there is constant breaking down of barrier and rebuilding again, and this keeps going on and on as long as the cell is alive or active. So the boundary is always subject to wear and tear, and especially the damage by, I would call it like oxidative damage, is too far for comprehension in terms of how frequently this damage happens and how frequently we are in need of this kind of free radical scavengers to keep the membrane in its proper integrity. Now this is one picture which explains about how this tocopherol is ultimately metabolized and eliminated from our body. So you see there alpha tocopheryl radical, and this alpha tocopheryl radical, which is basically alpha tocopheryl hydroquinone. So you see over there hydroquinone. So here alpha tocopheryl radical either could take the route of alpha tocopheryl hydroquinone, which eventually gets converted to alpha tocopheronolactone. Okay, so you see over there the alpha tocopheryl radical is also metabolized further to give rise to alpha tocopherinic acid. So you have alpha tocopherinic acid and alpha tocopheronolactone. So what are these compounds? So these compounds are traditionally mentioned as Simon metabolites. Okay, so Simon metabolites are the byproducts or the classical byproducts or end products of vitamin E metabolism in our body. So what does they contain predominantly? You find alpha tocopheronoic acid, okay, and then it's lactone, where the only known urinary alpha tocopherol metabolites, because if you notice it, we have already seen that most of the fat soluble vitamins, or almost majority of them, ADEK, is eliminated through bile. So they will be emulsified. Apart from going into systemic circulation, even elimination is through bile. In this case, if you are going to look at it, you see here the alpha tocopherol metabolites also do possess little bit of the action of vitamin E, but primarily we are looking them as degradative products. Okay, so the Simon metabolites are characterized by opened Chromand ring. So the Chromand ring is basically opened, and this provides the basis for the metabolite of vitamin E. So you look here where all this vitamin E components or activated products can find their way. So you see them in the proliferation. So they are pro-apoptotic nature, and you can also consider anti-proliferative effects. So recently people are studying a lot of vitamin E's use in cancer adjuvant therapy. So it's undeniable that people do benefit from these extra dosages of vitamin E, especially during chemotherapy or other inflammatory processes concerned with vitamin E related physiology in our body. So in that way you look at the anti-proliferative action. So most of the effects have been studied in animal model or probably in the human model as being closely observed for some major physiological response, but eventually you find them giving their way to probably additional functional aspects. So this is what we consider as the proliferation anti-proliferative effect, and then you have what we call it like lipid homeostasis, and the other one is their role in inflammation. So what is this trend is all about, vitamin E? So vitamin E, people are doing a lot of clinical trials to use this as an adjuvant in cancer chemotherapy and for people who basically suffer from inflammatory conditions. So you see over there, you always have, I would say, the vitamin E being used as a raw material for these kinds of physiological responses, and that's quite complex if you have to look at the lipid homeostasis. So you don't want anything to happen in long run, a damage or probably a repair in the cellular component, and the metabolites of vitamin E eventually find their way contributing in any of these functions. So we are going to look at, as I've told you in the earlier session, here we have protecting vitamin A and C, you add vitamin E. So it's like you could consider all these three vitamins, A, you could consider C, and the other one being vitamin E. All three, they are like interlinked with each other. Their function is very essential in terms of mutually helping each other in the metabolic role of antioxidation. So they not only preferentially get oxidized, preserving the more delicate and sensitive structures of the cell, but they themselves also undergo certain changes, as we have seen earlier, to really regulate the metabolic pathway. So now if you look at, this one we have already seen, enhances the alpha aminolevulinic acid synthase, and one more enzyme, ALA hydratase, is also involved in heme synthesis. So this way they could indirectly play a very important role in RBC physiology or RBC responses. So this is also a very important point you need to remember. this coenzyme Q. So coenzyme Q is a very important component in terms of electron transport chain. So electron transport chain acts as the hub of electron flow in generating, I would say, the power. Whatever that's been already generated as ATP has to be like recirculated, and there should be continuity of function, and electron transport chain is that extension chain which continuously ensures availability of energy, the metabolic energy, and this is like a kind of a powerhouse of mitochondria's, I would say, major generator. Okay, so you see here they are required by electron transport chain through cellular respiration. So which is one? That coenzyme Q. So coenzyme Q is a stabilizer, and vitamin E is a stabilizer of stabilizer. So it tries to enhance the function or leave alone function. It's essential for its normal, so biochemical functions of vitamin E. So we look over here as their role in the normal way by which you could consider how the other vitamins of this category like vitamin A and carotenoids are being absorbed and utilized by the system. Okay, so this we already saw in the previous session, and we are looking at how these free radicals basically cause damage in the liver, and now you can really check them up. Okay, so this is one particular area, what we call it like atherosclerosis. In fact, this slide what I've shown you is one from the recent papers which says that vitamin E has plenty of applications in atherosclerosis. Okay, so it is true, in atherosclerosis the plaque formation on the blood vessels, capillary vessels, are so, I would say, in a level where there is definitely a dysregulation of the corresponding vessel, and this eventually leads to embolysis. I would say there would be a block and the blood flow to the vital organs would be affected. So atherosclerosis is one of the primary causes of coronary artery diseases and all cardiac related insufficiencies connected with blood vessel, I would say, anomaly. So it is useful, vitamin E is useful in the treatment of atherosclerosis. It's of major relief. So for people who are already susceptible to atherosclerosis, maybe because of, I would say, dietary habits, and there are many other reasons over there, I'm just cited one example, and the treatment would be to put them on a long-term therapy on vitamin E. Yeah, you do have specialized drug for dissolving the clot and treating the conditions, but they are all symptomatically given. So the long-term, I would say, remodeling of how the vasculature changes leads to atherosclerosis. If it's been done favorably, it's a different story, but here we consider or talk in terms of a pathologic note. In addition to that, you do have plenty of other examples, like you say nocturnal muscle cramps, then cataract, then mammillary carcinoma. So these are some of the therapies. So any inflammatory, so if you could take a borderline in all this, let's say muscle cramps, there is injury, you consider the cataract being the fibrotic formation of the layers of your eye. So in case with cataract, it becomes like really opaque, preventing the flow of normal movement of light. So vitamin E supplementation on a longer note would really help, because most of these structures present in human physiology have an inbuilt mechanism of restoration. So vitamin E is part of that crew of proteins and components which help in restoration of damaged or inflammatory changes happen to tissues and cells. So in an industrial note or in a fortification sense, I have hinted on this in the earlier slides, and in the previous one I even explained about glutathione peroxidase role in selenium metabolism and how vitamin E synergistically plays a role with selenium. So means what? Together vitamin E and selenium not only would mutually improve its own efficacy, but it would also help in more profound way of treating inflammatory conditions. Now you see over there, although it looks like a kind of a wild claim, but that's really true. So aging process is one of the metabolic side effects, if you could consider. So metabolic side effects that leads to, I would say, aging process. So if you want to like really fix up these issues, you need to have, I would say, mechanisms to really hold back the free radicals, which otherwise can cause a very systematic damage. So you see over there, the aging process and the cumulative effect of free radicals have to be abolished. In other words, they need to be neutralized before you could really even consider of how you are going to treat age related problems. Now vitamin E comes very handy. Vitamin E is one of the wonderful preparations or vitamins which is used in keeping the proper tone of the skin. So considering its viscosity, vitamin E is generally used in all these preparations, or probably body lotions and all the topical preparations, even to improve the consistency of the preparation, at the same time to deliver whatever I would consider the most essential functions. So you see over there, other trace amounts of vitamin E, garlic acid and alpha naphthol are all there together as a package which will prevent the rancidity of fat. So what do you mean by rancidity of fat? We have hinted on this in the earlier session. So rancidity is, I would say, deliberate or environmentally conditioned effect on food products. Okay, so what will happen in this case? The food product what we are discussing right now would be oils or fats. So these oils and fats contain a lot of fatty acids. Okay, so these metabolites, I would say, which are responsible for rancidity. So what will happen? These metabolites, when they go out of control, okay, they try to really create a lot of damage. So in rancidity, what happens? A simple fat, which is a triglyceride, is now broken down into ester and fatty acid. Okay, not only that, most of these free acids, polycarbonated or polyunsaturated fatty acids, you find them, these fatty acids, playing a major exposure to oxidative damage. So they just cleave into fatty acid and glycerol. So when that happens, the original oil over there will have some distinct aroma. I won't say aroma, it's a distinct, I would say, smell, which is an indication that the triglycerides or probably the esters are not doing well. In such situations, how we do it? In other words, I would say, if there is rancidity of oils, rancidity of important, I would say, dietary sources of butter, then to improve the shelf life, vitamin E is added as an official antioxidant. Now you have a very good reason to ask, so what will happen to this component or vitamin E, which is added along with rancidity preventing components? So eventually the shelf life of the product would be much better, and you can always consider you are just restoring back the way how the destruction or metabolic damage to fats and oils are happening. Now you look at vitamin E, you also find other components like garlic acid, alpha naphthol. So these are also added as part of the stabilizers of antioxidant. We go over here. This I explained in the previous session, and I've today explained in detail about different aspects of I would say vitamin E function mechanism. We are going to look at some more therapies, like how vitamin E could be useful in treating certain conditions. So before we go on with that, so I want you to look at this picture, which clearly depicts how damaged phospholipids can still be, I would say, made available for the source for inflammatory components. So you should always consider the cell membrane is the native source for inflammatory substances. Most of the inflammatory substances start from there. Now the point here is, when there is a damage or a breach in this bilipid layer, we actually consider this breach is initiated primarily by, I would say, the pathogens which are trying to find their way inside, or it could be simply of mechanical reasons. So in any case, it's not only removing the pathogen from causing the disease or complications, but it's also about directly looking at how vitamin E could preferentially protect these higher components. So in our case, the focus of study would be the membrane. So the membrane, being a bilipid or phospholipid layer, constantly provides inflammatory supports, inflammatory components. So vitamin E would actually help in, through its antioxidant function, on these damaged phospholipids, because the free radical as part of the chemical reactions basically add on to the phospholipid damage which is already there. So here you find vitamin E goes over there and would scavenge and remove all sorts of unused, I would say, components. So when you say unused components, I mean to say those components which have some specific role to play, and if that particular thing is not initiated, they remain. So you see over here, vitamin E is basically bringing up a kind of a revival of all the damaged phospholipids. So eventually, when treated in the right amounts, so vitamin E would definitely be useful as an antioxidant compound. So here we have seen this, and now we are going to look at some of the side effects caused by, I would say, vitamin E deficiency. So to summarize, we have elaborately seen on the mechanism of action of vitamin E, and we have also looked at how vitamin E would play an essential role in oxidative damages and oxidative stress, and how you can really use vitamin D in therapy. Thank you.